Okay. Hello everyone. So this is the lecture on gene regulation. So we have talked about how the product being processed, being produced, right, from the very own genetic material, DNA, for example, and then it's been translated into the product, protein that we have discussed earlier, and it express as in the product. And after the expression, we will look into how we're going to regulate those expressions. All right. So this is all about, from shifting the whole idea of regulating, okay, the whole regulation to genetic uh, engineering. So you are engineering it, okay? Now, the whole idea here is the system. It's an operating system. The genome, of course, is not just a library, and it is an operating system. So this is what I meant, the operating system that we're going to discuss in a bit more on what the product, okay, from the very initial material, which is DNA. We're going to talk about how the system is controlling it, is regulating it. And then it move on to, okay, generating those information, the language that we would like to extract it, the coding, and, but we have two main system here, which is the prokaryotic and also the eukaryotic. So we're going to talk about that. And then how are we going to look into those codes, those coding? How are we going to not to modify, not to manipulate, but to engineer it? So biotechnology comes in. Okay, so this is the whole idea of, this is the master switchboard that you're going to hear the next few slides on how the regulation the gene regulation then eventually turn into genetic engineering. Okay. All right. So this is something, the image that showing that it's a kind of fish, right? It's a four-eyed fish. So what this whole idea here shows that uh, this fish has the same DNA, all right, exact same DNA, but it has different function, right? That means every cell contains the exact same DNA or this of it, but for example, the area of the upper eye express gene for example, the function for air vision, while the lower eye optimizes for water. So, but eye is exactly the same gene that, in the system, but what is controlling it that translate into the respective function? So this is how the regulation of the gene expression comes into picture. So this is the idea of differential gene expression, where it is the process where specific genes are turned on or either off, later we're going to talk about it, to allow cell specialization and energy conservation. Now, this is, I would like to, it's really important, as I always mention in the lecture, very much important for us to understand the terminology, the vocabulary of anything that we are going to talk about next. So when we talk about control, sorry, when we talk about control, the vocabulary of the control is great important. For example, you were going to hear about housekeeping gene, constitutive. This means that constantly expressed in approximately all cells, structurally maintenance, and on the other hand, something that you always will hear, regulated genes. So either is upregulated or downregulated. Upregulated meaning the level of these genes got risen up, and downregulated, it all falls based on respective needs. So in this regulation of gene, you have two areas or two types, inducible or repressible. So when we talk about inducible, eventually we have the idea by default, inducible, it is being switched off. Okay, by default it's off, but we need to turn it on by a trigger. We must on it. Repressible is by default is on, so we have to turn off by a trigger. Now you have to ask yourself the question, why we need to regulate? It is simply to conserve energy and space. Cells only produce what they need. So just imagine that there's no regulation, there's no control of the expression of gene. So things might got really out of control, okay? This is what I meant, okay? Like a DB board in the laboratory, in your house, a lot of control need, but you need certainly a lot of understanding when you look into this, okay? For example, the cockpit in the airplane, really important for the pilot to really understand each switches that the functionality of each switches and what will happen when it's turned on and turned off, right? So it's quite the same concept. So yeah, this is just for you to really read alongside what is gene regulation, is how cell control, which genes out of many genes in its genome are turned on. Turn on meaning you have this express. So that's why just now when I mentioned inducible is express, and then when turn off, it is more to repressible, right? So so the whole idea is to regulate the function in your body where you have so many expression of gene. And this is just a cartoon to talk about how different cells have different genes that been turned on and also been turned off, right? And of course, the factors that affect gene expression, a cell's gene expression is certainly a pattern is determined by information from both inside and outside the cell. For example, inside the cell, the protein inheritance from its mother, whether the DNA is damaged and how much ATP it has, and outside the cell, chemical signals from other cells, mechanical signals from extracellular matrix, the nutrients level, and so on. And we talk about how the cell responds to it, chemical signals, and so forth. And the whole idea, we are talking about gene regulation in both systems. Okay, later I will show you the difference of these prokaryotes and eukaryotes. But most importantly for you to capture here is prokaryote transcription and translation occurs simultaneously in the cytoplasm, and regulation occur at the transcriptional level. On the other side, the gene regulation eukaryotes basically is regulated during transcription and RNA processing. I'm sure you have actually, we have actually talked about that gene expression, okay? That during transcription and RNA processing, the regulation takes place in this area, which is in the nucleus, and during protein translation. Further regulation may occur through post-translation modification of protein. And certainly, you know, this is a table that should show what the difference of the regulation of gene expression of prokaryote and eukaryote. And the one that I highlighted need your attention, where the gene expression is regulated primarily at the transcriptional level. So gene expression is regulated at many levels, of course. And then we talk about later epigenetic, transcriptional, nuclear shuttling, post-transcriptional, and so on, right? Now, the very first system, okay, is the operon that we're going to look at. Operon, which is the whole idea. I give you another analogy to really understand this. It's like, you know, in a dance class, okay? So the nature's genetic conga line, operon are the ultimate party organizer. Okay, it is, you know, the host, making sure all the right genes show up and dance together when the beat's strong. Okay, hope you get that idea. Okay. So who are the VIP lists? Who are those? The key player. Well, someone called the promoter, which the promoter is the bouncer of the gene party. This promoter is the one that deciding who gets in and when, right? And then someone called the operator, of course, the DJ at the booth, where the music, which refers to the gene expression, can be turned up or down, right? And of course, the structural gene. So those structural genes are those dancer. Okay, the dance crew always moving together in a perfect scene. So I hope you get an idea by having another analogy, having another mode for you to imagine and to relate. So the whole operon is like a major genetic conga line. Okay, so it's just one conga line, and respectively they have their key player, promoter, the operator, and the structural gene. Now, what happened during the party? Okay, so there are two types of party that we're talking about, the surprise party and of course the after party. So when during the party, surprise party, of course, is happening loud. So usually quiet, but when the right guest, so when the inducer comes in, shows up, there you go, bam, the party starts. So you get really loud because you have someone called the inducer around. So example, we talk about it. If we relate to the inducer operon, is the lag operon. It's all chill until the lactose crashes the party. So this is, we're going to look at the mechanism later by giving you a flavor and the idea first. So what happened after the party, which we best refer to the repressible operon. That means it's over, but what's left? Okay, so the party always on until the party pooper, which someone called the repressor comes in and send everyone home. The trip operon is the repressor operon. Okay, so lag operon is inducible. Operon is the happening party, and after party, we always refer to the trip operon. When trip operon is always referring rocking all night until triple fun says lights out, right? So giving you the answer, giving you some idea. So you have a few key player, which is inducer, repressor, okay, and so on. So it's really important how the party gets regulated. Just imagine the party is not being controlled, so things might get very ugly in the end. So when you control, during the control or regulation, there's two types, the negative control and also the positive control. So negatively, like having a grumpy chaperone who stops the fun when they show up. So that's a negative control. You control in a very bad way. Positive control is that the one friend who gets everyone hype and dancing. So you will have fun, but in a positive way. Okay. So it is pretty much in control. Why operon are the life of the party? They are the ultimate multitasker, coordinating multiple genes like a pro-party planner. They help bacteria adapt faster than a chameleon in a disco ball factory. They are energy efficient, no wasting ADP or unnecessary protein production. Now, let's go into business. So this is what I would like you to really have to look at it. So when we remember that the surprise party, we always refer to the inducible lag operon. Okay, inducible lag operon. These are usually off, but can be turned on when a specific, remember, the inducer comes in present to activate a repressor. Okay, because the system is off, by default is off until the inducer, where the inducer, which refer to the lactose, right? The inducer is the lactose to inactivate the repressor. So negative control where the repressor blocks the bouncer, the promoter. Okay, so this is the conga line that we mentioned just now. We have promoter, operator, and these are the structural gene. Yeah, promoter, operator, and also the structural gene. So when there's no presence of lactose, the inducer, therefore the repressor are active. So it is just keep on turning off. There's nothing expressed. So, but the inducer arrives, which is the lactose, binds to the repressor, okay, making it fall off. So it results catabolic enzyme are produced only when the food is available. So this gives importance, very different. The regulation simply have this kind of mode. Okay, later we go a little bit deeper, but this gave you a very surface idea. Now, on the other hand, I just simplify the idea so that you can absorb better when you have a little bit more wordy the next few slides. So the after party is the repressible trip-operon. So the system is always by default is on, but can be switched off when something called the co-repressor bind to the repressor protein. Okay, until there's too much product. So by default, it's on. So the cell builds up tryptophan, a lot. So when the feedback inhibition comes in, kicks in, excess tryptophan acts as co-repressor, and therefore tryptophan activates the repressor, turning the system being switched off. Okay, so it being blocked and being switched off. So the anabolic pathway stops to save energy. So this is more to the repressible trip-operon. It's a repressible operon, which we refer to the trip-operon, which the trip-operon is always best referred, is always on by default, but can be switched off when the co-repressor binds to the repressor protein, which then block the expression, okay, of it. So a very quick recap: the eukaryotic, okay, the control via the separation, eukaryotes and also the prokaryotes. Eukaryotes, of course, is huge. Okay, it's like I mentioned, and the prokaryotes, like a studio apartment, is smaller. So prokaryotes, there's no nucleus, process occurs simultaneously. Regulation is primarily at the transcriptional level. Remember that? And eukaryote mentions massive here, compartmentalized, it's huge, and the regulation often happen during the transcription and also the translation level. So transcription obviously happening at the nucleus and translation happening in the cytoplasm, right? Okay, so this one is just something for you to read, like what we have discussed earlier. Okay. Yep. So this is not more. This is more too wordy. So you can actually read this as well for better understanding. But it's the same concept. Okay. So operons in the prokaryotes. So these are the conga line. You have the promoter, operator, and the structural genes. Okay, and then for the regulation of the metabolic pathway as well, we have the trip-operon. Okay, this is how it regulates for an excessive production of tryptophan, for example. So that's why the feedback inhibition kicks in, and then it will actually starts the regulation, can be switched off. So it binds to the repressor protein, which blocks the RNA polymerases and so on. Okay. So this all, the next few slide here is more, you can actually read, but as what I have explained earlier, it's the same. Okay. So yeah, so this is how the tryptophan or the trip-operon works. Okay. Repressor is more to for you to understand. All right. So in short, repressible and inducible operon, there's two types of this gene regulation. Negative gene regulation. Remember that the first slide that I said that even regulation, you have repressible and inducible operon, and then how to regulate, you can have a regulation, negative regulation, and also positive. So, but when we refer to repressible and inducible, it is best referred to the negative gene regulation. So as I mentioned, repressible operon best refers to the trip operon, and the inducible operon best refers to the lag operon. Okay. All right. Now, if you realize that in the lag operon, as I mentioned, is the inducible operon as well. When lactose presence and therefore the repressor inactive and operon got turned on, and therefore there's a lot more lag being produced and so on. So the regulation mechanism here, the lag operon employs two primary regulation mechanisms. Okay, of course, I have mentioned it actually falls into negative regulation, as same with the repressible operon. In absence of the lactose, the lag repressor binds to the operator, preventing the transcription. When lactose is present, it binds to the repressor, causing it to release from the operator and allowing transcription. At the same time, for the lag operon, it also carries in the positive regulation, where the scenario when the glucose level are really low, cyclic AMP, the level rise and bind to the catabolic activator protein like the CAP and form a complex to the CAP site, enhancing transcription of the lag. So it's either negative regulation or the positive. Positive is more, it enhances even greater, okay, but in a different mechanism. So positive and negative regulation is very important, okay? Because positive control or positive regulation, you need activated protein like CAP stimulating transcriptions and so on, right? Okay, so this is a negative, this is a positive, and this is a little bit more for you to look at when glucose level increase. This is a positive regulation. When you're talking about gene regulation, there's two types, negative and positive, so you can explain differently. So when positive, it enhance greater, okay? So you can give the scenario as what we discussed earlier is the glucose level, okay? Now, on the other hand, is on the epigenetics, right? So epigenetics, if you ask, when you coverage control access to DNA by modifying how tightly it binds around histone protein, it actually usually adding or removing chemical flags on histone temporarily alter the chromosome structure without changing the actual nucleotide sequence. Because this whole epigenetics, it actually is basically a study, okay, in how it's being organized and so on. So if we look at the chromatin packing, DNA winds around histone protein like beads on string. So the mechanism is very straightforward, something called the methylation and also acetylation. So methylation is like the lock, tighten and winding. So it leads to gene silence. Acetylation is the key where it loosen winding, where gene accessible. So the house rules is heritable changes that do not alter the DNA sequence, only how it reads. So I also discuss about heterochromatin and also the euchromatin. Okay, so this is how it looks like. Yep. So this is whole the epigenetic changes to the chromatin may result from the development, environmental chemical, it can result, those stimuli, okay, drugs, aging and diets, therefore some additional methyl groups and therefore changes histone a protein around which DNA winds to compacting and gene regulation. This whole methyl group, when in addition the whole epigenetic changes that the abnormalities, for example methylation or acetylation, the modification of histone tail, therefore alter the whole spacing nucleosome and change the gene expression, and this certainly leads to cancer. Those abnormalities in the gene that will results in cancer, autoimmune disease, mental disorder, and diabetes, and so on. But if you ask the question, are these changes permanent? The changes that occur to the histone protein and DNA do not alter the nucleotide sequence and are not permanent certainly. Instead, these changes are temporary, although they are often persist through multiple rounds of cell division and alter the chromosomal structure, open or closed, as needed. Right? So this is some of the technology, like how the multicellular eukaryotes have non-coding DNA. There are genes which do not code for protein, like transposon. Okay, a researcher named Barbara McClintock once said that no two plants are exactly alike. They are all different, and as a consequence, you have to know that difference. Uh, well, she said that I start with a seedling and do not want to leave it. I don't feel I really know the story if I don't watch the plant all the way along. So I know every plant in the field, I know them intimately, and I find it great pleasure to know them. See how passionate a researcher can be on her work. So Barbara McClintock, uh, you know, do a lot of research, certainly using the transposon and identifying something called the jumping gene. Okay, she's also a Nobel Prize winner, 1983, in physiology and medicine. You can read more about Barbara McClintock. And what she's trying to look at here is the DNA sequence that can change its position within the genome, and this is called the jumping gene. And of course, there's two ways of how the jumping gene works: insertion and also the complex transposon. Okay, so this results that the insertion sequence consists only one gene that codes for transposase. Transposase move sequence from one place to another can cause mutation if transposon occur in exon. Remember that I mentioned exon and intron. Complex transposon on the other side, where insertion sequence and extra gene, for example, gene for antibiotic resistance or seed color. Seed color, so for example, like the corn. Okay, so this is a very just a whole diagram that can simplify her work. Well, you have this the genome, you have the transposon. So when doing the transcription factor, okay, this actually much relates to the transcription factor, where to initiate a transcription, general transcription factor and RNA polymerase must bind to a promoter region. Okay, promoter region, which is called, refers to the Tata box, while other regulatory transcription factor binds to the distant enhancer region to increase or prevent transcription. Okay, so transcription factor, where the protein required for RNA polymerase to find the start line, which is the Tata box. Enhancer is a system control element that loops DNA to boost expression. So non-coding DNA here is probably around 98.5% of the genome, includes regulatory sequence, and therefore that's what Barbara McClintock research about the transposon, which is the jumping gene. Okay. So something to talk about, something to think about. Why do cells evolve? Gene regulation. Why is it important as well? Right. So yeah, this gives you some clue, but you can have more information. It helps them respond to viruses. It saves space and energy since they only create the protein they need. Epigenetic regulation prevents faulty DNA from being expressed. Okay, these are some of the quick checkpoints that you can look at. Now, we have reached the second half of the lecture, which we're going to talk about DNA technology. So DNA technology and genetic engineering involve pretty much direct manipulation of the gene for practical purposes, or how you're going to revolutionizing the biotechnology process. Okay, so you really know that how it's being expressed, how the gene being expressed, how it's being controlled, and then when with the knowledge you can apply it into the technology which is called DNA technology. So of course there's a lot of cool techniques that you can look at. many area. So yeah, this slide actually, if in the lecture hall, I can just ask you whether have you really watched this kind of movie, because these are first few of the movie that related to DNA technology. Okay, so if you have watched this, then you can relate and tell. So, well, of course, what are the main techniques and application of biotechnology? There's many, okay, as a core techniques. For example, from gene cloning, PCR, um, gene and electrophoresis, gel electrophoresis, DNA sequencing, and so on, okay, southern blot and everything. And certainly these techniques are really useful for a lot of application, for example, agriculture, medicine, environmental cleanup, and so on, okay. So if you look at it in general, okay, so it is actually used as a tool. Okay, it's a toolbox. It's a toolbox there where the main objective is to manipulate organism or their genetic component to make useful product. For example, we're talking about recombinant DNA. This is how it happened, combining sequences from two different sources. For example, jellyfish glow gene or the pig DNA. So you combine it, so it is recombinant DNA. So the toolbox is you cut and paste and then you clone it. So you refer the photocopier, like you use the tool PCR, and how you're going to sort it, you use the gene and electrophoresis technique. And how you're going to read those has been separated is the reader, it can actually provide you the DNA sequencing. The DNA sequencing or the method DNA sequencing that needed, that for example, the dideoxy chain termination method to use to read the exact DNA sequences, it uses modified fluorescent tag nucleotides that randomly stop DNA synthesis, resulting in fragment, verifying or varying lengths that can be read by laser to determine the genetic code and so on. Okay, gel electrophoresis is some techniques that based on separating the DNA fragment based on the size, shorter fragments to travel faster through the gel microscopic holes, right? And can I say, okay, the PCR is the polymerase chain reaction. So this technique rapidly amplifies, okay, specific targeted segments of DNA. It utilizes the three-step cycles: heating to denature, cooling to annul primers, and extending, with the heat stable enzyme called the tag polymerase. So you need a few chemicals to do that. So to exponentially grow and copy the DNA, right? So these are the whole idea. If you're talking about cloning, of course, to work directly with specific gene, normally a researcher will create identical copies. Okay, so you clone it by inserting a foreign DNA into the plasmid. For example, like these two gene that we combine, the gene and the pig DNA. So restriction enzyme, okay, act as a molecular scissors to cut the DNA at specific restriction site, creating a sticky end, and then the glue together by an enzyme called the DNA ligase to form a recombinant DNA. Yeah, this is how it works. Yeah. Cut and paste, restriction enzyme, and cloning. So act as a scissors is a restriction enzyme, cut the DNA at specific sequence, for example, Georgia Pro and so on. Just an example, okay? So what acts as a glue? The staggered cuts create sticky end that bond with foreign DNA. So ligase seal the bond. So the plasmid, for example, the bacterial DNA acts as an actor, and resulting the recombinant bacteria that clone the gene every time they divide. Now, when we talk about just now, the photocopier, as I mentioned just now, involve a few cycles: denaturing, annealing, and extension. So this is how, and you need a tag and polymerase. Withstand the heat. Tag polymerase, you can buy it commercially now, derived from the hot spring bacteria. Right, and this is a gel electrophoresis that I meant just now. It's in the electric field, it moves towards a positive electrode. The gel acts as propulsive, and then small fragments move faster and far, large, therefore get tangled and move slowly. So So this you can see. So after this step, how are you going to break the code? Yes, you put it into the Saga sequencing, okay, the machine to read what does that mean, okay, the sequencing. But of course, you need the fluorescent or the fluorescent tag or probe, okay, that actually can modify the nucleotide, stop the chain at specific point. Laser fragment is sorted by size, and the spectrum can show a graph color peak corresponding to the exact sequence of the bases, right? This is an example, the Dolly the sheep. Okay, in 1997, Scottish researchers announced the birth of Dolly, a lamb cloned from an adult sheep by nuclear transplantation from a differentiated mammary cell. Dolly premature death in 2003, however, as well as a arthritis, lead to speculation that her cell were not as healthy as those of the normal sheep, of course, because she had manipulated it, abnormalities happen. So possibly reflecting the incomplete reprogramming of the original transplanted nucleus. So application of the DNA technology, of course, affect our life in many ways. As I mentioned, there's now one reason forensic, agriculture, and animal cloning, environmental cleanup, and so on. Okay. One the example is a human gene therapy, alteration and afflicted individual genes. Okay. You can use it in the forensic. So DNA testing uses genetic marker called probably the STR, short tandem repeats, to create highly unique genetic profiles that can positively identify individual. Okay, for example, in crime scene, right? You can, or for criminals, can certainly use this technique. If you talk about the environmental cleanup, genetically modified microorganisms, for example, the bacteria, okay, can be engineered to extract minerals or degrade toxic waste, such as oil spills. For agricultural application, the whole animal can be cloned through the nuclear transplantation, for example, and where the nucleus of the unfertilized eggs, for example, talking about the fish, in the nucleus, okay? No, when we talk about agriculture, like the, the Dolly the sheep, okay? And let's say the fish, so it's pretty much they look very different. Okay. But with all that, yes, it's very useful to us human and of course change our life. But something that raised a concern where safety and ethical consideration comes in about, where those powerful application of genetic engineering certainly provoke significant ethical and safety questions. There is ongoing concern about potential long-term health effects of eating, for example, GMOs, genetic modified foods, GM foods, the possibility of those transgenic protein causing, you have allergy, okay, allergic reaction, the risk that can transfer gene could create super weeds that can harm local ecosystem. It really disturbs the ecosystem, okay, biodiversity and all certainly results in the national and international guidelines to really look at all these abnormalities, all these guidelines needed to strive to regulate. So must control. You can apply, you can use so powerful application, DNA technology application, but certainly have to have the regulation to have this stricter and safer practices. Okay. So this is just an example for you to look at genetically modified cattle treating Ebola, right? And also this just want to share with you. Scientists have concern about some users genetic engineered food, okay? Possible long-term health effect, as I said that, right? And disturb our ecosystem. something to think about, or this question can help you to think and look for more information. What are some of the ethical implications of modern biology? How should scientists address this concern? Because otherwise things will go very ugly. Now these are those images on animal, but how about on human? Okay, just imagine, right? So lastly, something for you to take home. Take-home messages. Bacteria often responds to environmental regulatory change by regulating at the transcriptional level. Okay, concept of operon and types. We have two types of operon, reproducible and inducible. And of course, we have two types of gene regulation, positive and negative regulation. Eukaryotic gene is regulated at many stages. Okay, as you can remember, DNA sequencing. Also, we move on to this DNA technology, where certainly important involve many direct manipulation. Okay, you manipulation of genes for many practical purposes in the biotechnology field. So with that, I think this is the end of the lecture on gene regulation. So I hope you can read more, and later I will share some question on this lecture, and as soon as you have completed this recorded lecture, you can actually try to attempt those questions to test your understanding, right? Okay, thank you all. Bye now.